All right, so hi everyone. I'm Edward Lau from UCLA, and as Vincent mentioned, our regional speaker couldn't make it today, so Brian and I are going to give a brief update on one of our projects and recent publications on how to access and analyze protein turnover data on Synapse and ProteomeXchange. So I will start by summarizing what some of the recent progress, with the protein turnover data set that we acquired, and also sort of tie into what we're working on with data dissemination mechanisms and promoting open data access and collaborative analysis. And I'll give some backgrounds on what we're trying to do to share both raw and processed data files on these repositories. Basically, hopefully serving, having this serve as a model for other data sets going forward. And then I believe Brian will give a brief demo on how to actually access the data sets and what you could do with them. So starting with a bit of background and motivation on the data set. So we have acquired a large data set of proteome dynamics, which contains the information of the turnover half-life of about 3,000 proteins and the abundance of about 8,000 proteins in 6 genetic strains of mice using a metabolic labeling, high-resolution mass spectrometry, and kinetic modeling method. And we are trying to make this data as widely available and accessible as possible before we we try to interpret the biological results. So there are a number of reasons why we want to do that, and I don't think I need to go into too much details to this audience why you should share your data. But for me as a biologist, what I think is as data acquisition in proteomics becomes more and more mature, oftentimes the room for innovations and breakthroughs are going to be increasingly coming from the people who could best make sense of the data rather than people who could generate the most amount of data. And data interpretation increasingly is going to require very specialized skill sets in data science. So in order to kind of get the most out of the data for everyone, then the data should be made available to people who could actually develop new methods and help biologists look at the data. So there are a number of reasons why we think this is a special and common data set. There aren't a lot of protein dynamics data available, especially when it concerns data that are measured from in vivo animals rather than just in vitro cell cultures. So it would be good to for people who work on, for example, understanding the relative contribution of the regulations of protein expression to have access to this data. And in general, having data and code available help promote transparency and reproducibility in publications and studies. And also, we think having this data descriptor manuscript as a companion article to this publicly available data set will help promote the visibility of data and also promote open science. So, as I mentioned earlier, a data set is publicly available, and we have a data descriptor manuscript that came out since the BD2KPI meeting at EBI in February, and it's on this website here that you could access through the address. It was also featured on the front page of Scientific Data. So if you go to the journal front page, you're directly to the article. So what is a data descriptor? According to the description on the data science website, data descriptors data descriptors on Scientific Data are open access articles for publicly available data sets. They're actually aiming to help others reuse the data rather than presenting an interpretation of a hypothesis per se. So it generally breaks down into a couple of different sections. It will contain some background and summary on why this data set is maybe valuable, and also contain very detailed methods on how the data were acquired. Then you'll have a description of the data records where you could find the data, and also you'll contain some technical validation information about the technical and statistical rigor of the data that may not otherwise be very prominently featured in a biology manuscript. And lastly, we have a discussion on usage notes on how we envision people could make use of the data. So I'll just go over a few of these categories in the manuscript, starting with a background and summary session. This would be, in a way, this would be a recap of what we presented in the previous rounds and also during the PI meetings. I'm not going to go over it in a lot of details, but basically the motivation is that a steady state proteome will respond to different stimuli, whether it's a drug or a disease, to reach a new steady state that supports new phenotypes. And it could do so either through an alteration in RNA expression or some kind of changes in the protein life cycle, such as the synthesis or degradation rate. And it's a topic that is under debate, what the relative contributions of RNA protein synthesis and protein degradation are in this kind of process. There's some initial studies that found there was very poor correlation between RNA levels, and the very poor correlation between measured RNA levels and also protein levels, which was taken to be interpreted as showing that RNA doesn't really control protein abundance that much. More recent studies paint more complex pictures, but the problem with some of the existing works is that they're mostly done in vitro, where protein degradation mechanisms could be very different. In a plate of cells in vitro, the half-life of proteins are usually in the temporal regime of, in a matter of hours rather than weeks, which would lead to an underappreciation, I suppose, of the contribution of minor changes in turnover rates. So we're interested in trying to understand protein regulation and transcript regulation and protein dynamics in a more physiologically relevant in vivo model. And this is what this data set is about. We have a quite detailed method description in the manuscript, and here I'm just showing a schematic from one of the figures in the data descriptor manuscript, basically showing on the left-hand side here how we perform the metabolic labeling in disease model, and followed by a simple workflow on how we extract and separate proteins and then analyze it by mass spectrometry. And at the end, you see this little blue arrow here. We acquired this data set, and and this data will be analyzed through a data analysis and data dissemination workflow that we depicted here. And with the data analysis, we basically identify the proteins that are present in the mass spec data with ProteoZip, and then we calculate the half-life of proteins using Proton. And this figure also shows at which point during the analysis we would disseminate both the raw and processed data files. And we have two types of files with raw data. On the top here, we're disseminating them through ProteomeXchange and then Pride. And then on the bottom here, with the turnover rate data after technical validation, we are disseminating them in Synapse. And we also mention a potential use case in this figure here. So the article also contains a data record. So these are figures that usually might not be given a lot of space in an actual biology manuscript, but I think they're very important in showing people the shape and the quality of the data. So we have figures that show the correlation and reproducibility of the data among the 6 strains, and also the distribution of turnover rates in each sample. And lastly, we also have panels for data validation to show potential users of the data, what the quality of the data are, and what are maybe some of the caveats into interpreting the results. We have panels that show the cuff ratio of proteome for different measured parameters. We also show some of the example fitted kinetic curves on panel B here, when the goodness of fit are in different values within the range of acceptable R-squares. We have the general distribution of goodness of fits and relative standard errors within the data set, and we're showing the intraprotein variances. And also on the bottom here, we show that different peptides within the proteins have quite similar and reproducible turnover rates, which is what you would expect if the data quality were good. And And lastly, we sort of enumerate some of the potential use cases to maybe help people use the data and inspire people on how you could reanalyze the data if you're a data scientist, or you're interested in similar problems. For example, we recounted some of the use cases we could think of. The first one is you could just simply look up a turnover rate from this data table, right? You could retrieve half-life and abundance of a protein of interest. For example, if you're a cardiovascular researcher that's studying mitofusin and you're interested in knowing what its half-life is in the heart, then you could look that up. And you could maybe use, for example, proteins in the mitochondria. You could use the absolute half-life to calibrate some of the fluorescence timer that you're using to measure mitophagy, for example. Secondly, you could perform pathway analysis, because we have a rather large data set that covers maybe over 10 organelles and about 200 reactant pathways. And you could use this data to analyze different protein complexes, pathways and compartments. For example, if you're a basic scientist that studies the Golgi apparatus, and you could try to see if protein half-life within the Golgi are different from other organelles, or how they change during hypertrophy. Thirdly, we think that, because we made the mass spec data files available, then if you're an omics scientist, you could reanalyze the raw data. If you have a more sophisticated way to identify proteins in the future, then you could also find out the half-life of those new protein sequences that you could identify. For example, if you have a proteogenomic database, then you could identify proteins that would indicate single amino acid polymorphisms and so on. And lastly, we have a complete data set on protein kinetics that used for further method development to understand protein half-life. For example, if you're a data scientist that's developing a new kinetic model or new software to analyze protein turnover data, then you can use this data set as a reference to validate your results. And then, so this is my last slide. Basically, and I'm going to hand it over to Brian after this on how you could navigate the data set. But basically, as I mentioned, we've uploaded data to different places. Pride and ProteomeXchange will be the main repository for the mass spec files. We have over 1,500 raw files on a project on Pride. But there are a lot of files that you can store on Pride and ProteomeXchange. For example, the semi-processed data and the parameterized turnover rates and the processing scripts and graphs, and those are found on Synapse. And he also has a wiki interface that shows you where the data are and what the individual columns in the data table mean and how you could access them. And lastly, we have a step-by-step instructions and some sample code on the COPAKB website that shows people who are maybe not experienced with Synapse how you could access the data and what you could do after you download the file to look up, for example, the individual half-life of proteins, in use case one that I mentioned. So it's Brian there.
Yes, thank you, Edward. This is Brian Bleakley, and I'm going to basically briefly go over some of the use cases Edward mentioned in the previous slide and show you our instructions on the COPAKB website.
It's weird because everyone seems to be able to log in.
Okay. So if you come to our website, heartproteome.org, and you get through the splash screen, you'll see here we have a little banner announcing the Synapse protein turnover data set. If we follow that banner, we'll come here to a page with a series of step-by-step instructions covering different use cases. So the first use case I want to cover is the simplest one, to view the processed outputs on the Synapse website. So we'll follow the link here. The first thing you see is an introductory page. We'll go to the files section, and we'll go to the tidy folder. And the tidy folder contains the organized, polished output. And I want to download all protein K, K being the turnover rate. You also have to, if you want to access these files, you'll have to create a Synapse account. If you are not registered on Synapse, you can browse the structure, directory structure, but you can't actually view any of the files, view or download any of the files. Okay, once I download the file, it's a tab-limited text file, so I can drag it into Excel to analyze it. Okay. If I go to the Data tab and I go to click on Filter, it'll identify the top row as consisting of column headers, and then you'll have the option to filter and sort by each column header. So that's an easy way if you want to identify a particular protein. You can type into the search box. And you'll filter the results. And all these instructions are covered here on the tutorial page on hardproteome.org. So that's the way to view the experiment outputs. If you want to do some analysis, or you want to do some reanalysis of the original raw mass spectrometry files, those files are not on Synapse. As I mentioned, those are on Pride. So we can follow the link here to view those files. There's a brief description of the project. You can click on Download Project Files to get to the archive. You can download these files directly from the website. You can also use Aspera. You can use FTP. It'll take a while to load up the index. Here you can see there's an index of all of the raw files that are available. So you could download these one at a time if you wanted. On the hardproteome.org tutorial, we have examples of some commands if you want to programmatically download a large number or download a range of files. You can use a curl command to get this entire manifest of files. You can use a wget command to download a particular file or a set of files, or to download all of the files. Once you have downloaded all of the raw files, you can use a database search engine to identify proteins and peptides in those files. So you can use either Mascot, ProLucid, MaxQuant, or another database search engine. And we provide, in this paragraph, we provide some recommended parameters, search parameters, if you are going to do that. And for the final step, you can use ProTurn, which is the software we originally used to calculate the protein turnover rates. ProTurn requires a particular directory structure, which we detail in the ProTurn documentation, because each database search result file and each MZML file needs to be associated with other files of that time point to be able to perform the analysis. So you set up a folder structure with your search results in it, labeled based on the time point, and the MZML files in separate folders organized based on the time point. And because you'll need to use intermediate software such as Proteome Wizard to be able to convert from the raw files into the MZML file to be able to analyze them in ProTurn. If I load up ProTurn, the process is pretty simple. You select your file format, you select your data directory, The data directory you're selecting is the root directory that contains both the MZML folder and the database search result folder. So I'll take and then you select all your parameters and you start the integration step. This integration step could take an hour or more for a large data set. Once you're finished, the next step is to fit the kinetics curves. Again, you fill out your parameters and fit the curves. And this kinetics curve step is very short. And will only take a few minutes.
Can I ask you a general question now while you're in the middle? Can you hear me?
Yes, go ahead.
So if a user or customer, or we want to call it, is trying to identify a specific data set, so is it the file descriptive enough, or does it need to download like the metadata first and say, I don't know, I want all the hearts of this specific knockout and the time course? I don't know all the data that you have there, but how does an outsider user approach like to ask a specific question, download the exact data that he will need to do that?
I'm not quite sure. Could you give an example of the specific question?
Let's say I would like to know on if you have the data on a specific knockout mice, and that you have done all the kinetic and that all the labeling, and if you have a time course of that, what are the data files that I would have to download to look certain number of organs? Like, for example, you have liver, heart, and kidney. So I can see that, for example, that denomination of where it is the tissue, it seems to be in the file. What is the mouse type? I'm not sure if it is in the file name. If it is, for example, at what time in the stage of some information about the metadata. How can I search all your database, if you wish, of metadata for the data that are there?
The strain of the mouse is one of the columns here in the all protein K file, which is the overview file. So
that information is also on the scientific data manuscript. The data descriptor there actually also comes with a metadata file on the experimental conditions and strains and organs and time points. There are certain things like which protein is in there that you would need to go to Synapse in order to find out. But in general, you wouldn't need to do a lot of coding just to find out what data are in there. Most of the descriptions are either on the manuscript or the Synapse project. I don't know if that.
I have not read the manuscript, so my question may be naive. But so you provide a file that is a metadata files for all the data that you can search and identify what are actually the raw data that you need for your specific question that for your specific data that you want. Is that correct?
Right, that's correct.
Okay. All right. Thanks.
There's also Brian, if you could maybe also go to the table view on the Synapse project, you could also look up that information there, like without doing any coding. There's an index of all IDs and results, so you could search directly from Synapse using the query in the query box. That's one way of doing that.
Yeah, so here, yeah, you can see under the tables tab, as Edward mentioned, there's an index of results, and you can create a custom query here, and you can see the sorted by mouse strain, treatment, data type. Let's see. That's about the end of this walkthrough. There are a few other use cases here on the tutorial on hardproteome.org, and that's basically a walkthrough of how to access the files on Synapse and access the files on